Hi everyone! I'm Sanya Kuller. Welcome and I hope you're doing well. Greetings from the United Kingdom. Okay, that's my attempt for a British accent. So in this video, I'm going to take you through a step-by-step example of how to run the Polygenic score PGS catalog calculator on the DNAnexus platform for the UK Biobank genomics data, the genetic data. Now you might be wondering, well, what exactly does this mean? So let's break it down. The UK Biobank is a huge research study with genetic and health information from about 500,000 people. It is one of the most important data sets in medical research today. And so the big question that we're asking here is, can we use someone's genetic information to estimate his, her, or their risk of developing a certain disease or trait? To make things concrete, I'll be using Alzheimer's disease as an example. Alzheimer's is a progressive brain disorder that affects memory and thinking. It's one of the most studied conditions in genetics and precision medicine, and it makes a perfect case study for this tutorial. And what is the PGS catalog? So the PGS catalog is short for Polygenic Score Catalog. It's a free open resource that collects and shares polygenic scores, PGSs, that are published by researchers all over the world. And a polygenic score is simply a number that adds up the effects of many genetic variants, often thousands of them, to give you one measure of a person's genetic tendency or risk for a trait. For example, instead of looking at one single Alzheimer's-related gene, a polygenic score combines information across the genome to get a fuller picture of genetic risk. So why is this important right now? And why is this video about working with DNAnexus important in general? Well, because recently, access to the UK Biobank genotype data is moving towards the DNAnexus cloud platform. And it's happening really soon. And I know a lot of researchers and people out there are a bit overwhelmed and nervous about the transition because now a lot of you may have had it stored on your local computers, machines, that data, but now no longer will that be allowed. It's all moving on the cloud. And if you want to use the UK Biobank data for your own research, you'll need to know how to run any type of genetic analysis, using UK Biobank data in this DNAnexus environment. So I'm going to help you with that. And even though our goal is eventually using the PGS catalog calculator, I'm going to be helping you get more comfortable with using DNAnexus. So by learning how to use the PGS catalog calculator on DNAnexus, you'll be able to apply polygenic scores to large data sets like the UK Biobank, replicate published findings, and extend these approaches to new diseases, traits, or research questions. And what you'll learn in this video and in this walkthrough, I'll show you how to set up and run a polygenic score calculation for Alzheimer's disease. I'll explain the key steps from preparing the data to running the calculator and share some common pitfalls and troubleshooting tips that can save you time. And I'll provide examples and explanations that are general enough for other diseases or phenotypes that you want to study. But here is the best part. We're going to be hopefully learning a lot along the way. And even if you're completely new to the UK Biobank, DNAnexus, or polygenic risk scores, this video will help you understand how genotype files are structured and used, what is a polygenic risk score, and how it's calculated, how to navigate and run analyses on the DNAnexus cloud portal, and it's all sorts of things along the way, not just the risk prediction, but even working with the basics in DNAnexus. And also, more broadly, how genetic research connects to predicting disease risk, the genotype to phenotype link. So whether you're a student, a researcher just starting with UK Biobank, or simply someone who's curious about genetics, this video will give you both practical skills and foundational knowledge. And finally, if you find this video helpful, please remember to like, subscribe, and share this video with friends or colleagues who might benefit, and buddies as well. And I'd love to hear your questions and feedbacks in the comments as you try this out for yourself. And again, all right, with that, let's dive in. And again, this is things that I've been discovering from my work that I thought might be helpful for you. and I really just hope it can make your lives easier as well. And whether or not you're calculating polygenic risk scores, if that's your interest or not, this video will be very helpful for you in many different ways, and I'll be organizing it in chapters so you can go to the parts that you find are necessary for yourself. And next, I'm going to tell you what are some of the few of many things I hope that you may learn from this video. So again, I'm Sonia Kuller, and this is a little bear is joining me, and this is a little moose. They're joining me on this journey, and let's get started. And again, don't forget to reach out with any and all questions that you have. So in this video, as I was mentioning, there are a lot of things we're going to be covering, and as they say, to infinity and beyond. But what some of the things I'm going to be covering, this is just some of them, just to give you a little flavor, but it includes this and beyond. So like navigating the PGS catalog, gentle introduction to polygenic risk scores and genetic scores in general, how to subset BGen files for a list of variants that you have, how to convert BGen to BED files, a Plink1 format, how to merge BED files across chromosomes, how to upload files using DX Upload, how to watch your jobs using DX Watch, using BGenX and Plink2 tools, using the DX toolkit and the terminal, running codes using Bash and Python. We'll be doing quite a bit of that. Running PGSC calc tool on DNAnexus to calculate these polygenic scores. An overview of the different genotype file formats, just going around the research analysis platform, looking at some of the UK Biobank genotype data and the cohort browser, working with the Swiss Army knife toolkit and beyond, and just working with jobs on DNAnexus. So there's a lot more, but this is just a little flavor. So whatever you think you can get out of this, I really hope it will help you in some capacity. Okay, so just a little bit of a background on the UK Biobank data. So its cohort is around 502,000 participants as of the time of this video in 2025. The recruitment was in 2006 to 2010 at 22 assessment centres across England, Scotland, and Wales. And the age of recruitment was 40 to 69 years. And the geography and ancestry are primary residents of the UK, United Kingdom. And the majority is of European ancestry, around 94 to 95%, with representation from South Asian, African, East Asian, and other ancestries. And there's phenotypic data like questionnaires on lifestyle, diet, physical measures, imaging like magnetic resonance imaging like MRI, DXA retinal, like health outcomes. And there's longitudinal follow-up, that means over time. And it's linked to the National Health Service, the NHS hospital records, primary care, cancer registries, death records. And so there's a lot of genetics and omics data, like multi-omics, like different types of omics. There's genome-wide genotyping for around 96 million imputed variants. There's WES, whole exome sequencing for around 470,000 participants, and whole genome sequencing, WGS, for a subset. And the multi-omics data that are in subsets, like proteomics, like Olink, SomaLogic, there's metabolomics, there's microbiome. And the use case for this data is for large-scale genetic association studies, for discovering biomarkers for different conditions, for disease risk prediction. We're going to be doing it in our example for like late onset Alzheimer's disease, and for therapeutic target identification. So trying to find what could be potential avenues to try to ameliorate, to lessen the impact of certain conditions and diseases. So it's all really helpful for drug target discovery and for trying to find out potential cures for different diseases. Research is all like a stepping stone. And just for you guys to know, there are other cohorts with genotype information out there, like All of Us Research Program and the US, of which I'm a proud participant. Yay! Woohoo! So, as they say, diverse DNA data improves disease risk scores for more ancestries. So we're able to better predict, disease risk for individuals. So just something for you guys to keep in mind. So we were focused in this video on UK Biobank, and there are other cohorts that are out there, and more data is being made available. So I do encourage all of you, if possible, to try to contribute to these DNA sequencing studies and participate in these cohorts if possible. It'll really help make a difference, believe it or not. Okay, so this guy is as close to my little tubby as possible at this moment. So as you can see, that in the spirit of GitHub, the cat, that's my little orange dude, a little boy, Tubby. I'm his mom, and that's me here. So I'm giving an example for how we go through and do this analysis. And this is for my example for you guys for like late onset Alzheimer's disease and so on. It's just an example for how to do it. So I'm showing you some specific examples and walking you through it. And I will be posting the code on GitHub, so it's publicly available for you to follow. So it's for your reference, for reproducibility, for you to use it as a template. So please see the video description information below the video for details. And also PGS catalog calculator, the documents, they will shortly be uploading my code as an example for all of you. So when you check the website soon, that should hopefully have the tutorial that I'm also making for that for the team at PGS catalog calculator. So please be posted. But below I'll be pasting the link for the GitHub repository for you to follow along. So please do check that out. But yeah, the goal is that I'm walking through an example, my example, but I'm hoping that you can apply it for your data. It's just, in reality, it's a cartoon example. So you can apply it to your own data set and for your own analysis and adapt as needed, and reach out to me with any and all questions that you have. So that's the cartoon of us. This thought is really cute. And so yeah, please do follow along with the cartoon example that I'll be using, and reach out with any and all questions that you have. Adapt it as needed. It's just a cartoon example for you guys. And when you're running PGSC calculator, you can add more more parameters and other things as needed, and more advanced data sets. I'm just trying to make a cartoon of the real world examples that you may encounter. So in this tutorial, we'll be going through the following files here, as you will see, and I will be calling it the Sanya demo folder. It's saved on my desktop. We'll be starting with the PGS calculator _test.sh. Then we have these other steps that we'll be going through, simple starter.sh to create a set of folders as an example. Then extracting PGS snips.ipynb. It's a Python Jupyter notebook using Python to basically go through the score file that we have and create a bunch of text files, one for each chromosome, basically telling you which are the reference SNPs to extract for that chromosome. Then we'll be basically using that set of SNPs to extract for each of the the text files for each chromosome, and we'll be using the BGen files, like the genotype files on the UK Biobank portal. We'll be kind of subsetting those BGen files for the set of SNPs that we need, and then we'll be converting each of those subset BGen files to bed files using this step 4. Then step 5 will be kind of like merging all those like bed files for each of the chromosome, all those files into one set of files across those chromosomes. So one set of BIM, FAM, and bed files. And then we'll be running the PGSC calculator in step 6. And so we also have a sample sheet we'll be using for PGSC calculator, and the score file that we will be working with for which we will be computing the scores, the polygenic risk scores, which will be Alzheimer's disease. But these will be the files that we'll be working with. And I've been making them publicly available so you can follow along, and these are the files that you have to use to follow the tutorial. So yeah, we will be doing a lot of different things in this video, and here's a step-by-step process, you know, Magic School Bus style here. Welcome to Magic School Bus. Okay, so for the genotype to phenotype prediction, right? So I'm going to show how we run the PGSC calculator test. So that's a test one, just to make sure that everything is kind of working and that, like, you know, we can at least run the demo that they recommend. So we're going to run this. Then I will show how we can download a human score file from the Polygenic score PGS catalog. Then the first step we will be doing is creating sample folders in the DNAnexus platform, just to kind of get the hand of things, you know, get a handle over it. So yeah, this is our game plan, lesson plan as they say. And so we'll just be creating some folders. Step 2 is to extract the PGS SNPs, you know, from the score file. Then the third step is to subset the UK Biobank, the UKB BGen human genotype files for the PGS SNPs that we have for the given chromosome. So we're going to go through the score files, and you know, we have in step 2 already extracted the set of SNPs that we need for each chromosome, and then for the given chromosomes, because some chromosomes may not be needed for the score, which we will see. So we're going to subset the BGen files for just those SNPs that are needed for the score. So we're going to have reduced BGen size files, and we will convert these BGen files into the corresponding BED, BIM, and FAM Plink1 binary genotype files for the given chromosomes. And then what we'll do is, so then we'll have one file for each of the chromosomes, one BIM, one BED, one FAM file for each chromosome. And so we will be condensing them all so that we just have one set of BED, BIM, and FAM files. So that's what we'll do in step 5. So merge these Plink files across all chromosomes, you know, for the chromosomes that are there. And then in step 6, we'll run PGS catalog calculator on the DNAnexus platform for our data, for our genotype data, and for our score file. And then I'll just show some additional downstream analyses that we could do. But this is just an overview of what you can expect. And along the way, we'll be kind of covering different topics that could be helpful for you. And so there'll be these 2 checks that we want to do. The first is to make sure that we can log in to the DNAnexus portal, the research analysis platform, the RAP for the UK Biobank. And the second is to please ensure that you have enabled the DX toolkit on your local computer or your server, wherever you'll be storing your local files that you want to run on DNAnexus. Just make sure you have the DX toolkit enabled, please. In the spirit of you know, being in the UK and talking about the UK Biobank, I'm going to say, please like, please subscribe, and please share this video, and reach out with any and all questions and comments that you have. I really enjoyed making this video.